Welcome to today's Walls lecture. Please take your seat. We try to keep in time today because there is a lecture right after this Walls lecture. It is a great honor to introduce on behalf of the NIH Fellows Committee today's speaker, Dr. Witold Filipowicz. Dr. Filipowicz comes to us from Basel, Switzerland, where he has been a group leader at the Friedrich Miescher Institute for Biomedical Research for the past 28 years, and a professor at the University of Basel since 1997. He was born in Poland and graduated as an MD from the Medical University of Łódź before receiving his PhD from the Institute of Biomedical and Biophysics of the Polish Academy of Science in Warsaw. With his PhD studies about translation of RNA phage F2, he launched a very successful career and journey through the entire universe of RNA. This journey brought him first as a postdoctoral fellow to the United States, where he joined the lab of Nobel laureate Severo Ochoa at the New York University. He moved with Dr. Ochoa to the Roche Institute of Molecular Biology in New Jersey, where he started working with Alan Shatkin, with whom he kept a productive collaboration after returning to the Polish Academy of Science and establishing his independent research program. He was then recruited to the Friedrich Miescher Institute in Basel, but kept initially his lab in Warsaw. However, working behind and traveling across the Iron Curtain became more and more difficult, and he ultimately moved with his family to Basel in 1984. During his outstanding career, Dr. Filipowicz made seminal contributions to many aspects of RNA biology, including RNA processing, enzymes regulating RNA metabolism, the role of non-coding RNA in gene expression, and more recently, RNA interference, and the role of microRNA function in mammalian cells. With his most recent research, he shed light into the clinical relevant microRNA-mediated processes, ranging from host virus interaction to the light adaptation in the retina. For his work, he received numerous awards, and he is a member of many distinguished scientific societies and editorial boards. In recognition of his long list of achievements, he was honored last year by the International RNA Society with the Lifetime Achievement in Science Award. And his home institute for 28 years just held an honorary meeting, I think 10 days ago, with having all the important people in the RNA field present and as guests in Basel. Like many of you, I ran first across his name on the servers of the National Library of Medicine when I started working on microRNA. Soon after, I had the exceptional opportunity to meet him personally in Switzerland, and this introduction would not be complete without saying that his warm personality is as impressive as his scientific achievements. The title of his talk today is Regulating the Regulators, Mechanisms Controlling Function and Metabolism of microRNAs. Please welcome Dr. Filipowicz.
Thank you, Stefan. This is really embarrassing to hear such introduction, and feeling like a really kind of dinosaur of RNA research, but still active, not extinguished completely. So, although they were trying to extinguish me with this symposium 2 weeks ago, but I managed to survive. So it's a pleasure to be here. I've never been to NIH. I have had a lot of contact with NCI and Frederick, but I somehow happened never to visit the campus, and I'm quite impressed with the people here. Of course, I know many people, And I'm grateful that I could talk to them today, but also the campus itself, it's the way it looks. So as Stefan mentioned, I will be talking about mechanisms controlling function and metabolism of microRNAs in metazoa. I will spend about half of my talk on a function and mechanism of microRNA repression, and then we'll go into regulation of metabolism of microRNAs. And just to start, I would like to remind everybody that we are going through some kind of a paradigm change in gene expression studies. And until recently, we knew that about 1 or 2% of the human genome encodes proteins, and it was not clear what the rest is doing. And it is now clear that 90% actually of human genome is transcribed, but this small percent encodes proteins, but the rest encodes non-coding RNAs. And we are busy now trying to find out what those non-coding RNAs do. And this situation is completely different from E. coli, where 90% of the genome encodes a protein. So that probably explains the difference between E. coli and us. So there are many classes of non-coding RNAs, long ones like XIST or long intergenic non-coding RNAs, link RNAs, imprinted RNAs. A couple of them were known. Now there are thousands of them. There are also intermediate length microRNAs, exemplified by small nuclear RNAs and small nucleolar RNAs. We used to study those for many years. But then there is this number of small RNAs, 20 to 30 nucleotides in length, and I will entirely focus on one class, on microRNAs. And microRNAs are about 20 nucleotides long post-transcriptional regulators. They are encoded in a genome. They are transcribed usually as long precursors which fold to hairpins, which are referred to as primary microRNAs. And those are processed in the nucleus by a complex of Drosophila, the RNase 3 enzyme, into shorter hairpins. So the cuts are taking place here, referred to as pre-microRNAs, which are then processed by dicer in the cytoplasm, and one of the strands of this duplex is incorporated into microRNA, microRNP complex, also known as microRNA RISC, with AGO and GW182 proteins as a key components. And this complexes bind to the 3' UTR of mRNA and induce translational repression and mRNA deadenylation. And this class of small RNAs is active in animals, plants, and algae. About 1,000 or even 1,500 microRNAs may operate in humans. Most of the genes are regulated by these microRNAs, and they practically regulate any process people were looking at. And just to give you some introduction, some principles how microRNAs regulate mRNA targets, their sites are usually spread through kilobases of 3' UTRs. 3' UTRs are often longer than coding sequences. They can be 10 kb long. And their sites are usually enriched near the termination codon and poly-A site, present in AU-rich regions. And the key element, which is important for recognition of microRNAs, but there are exceptions to that, is a so-called sequence, a seed sequence, which is a position, nucleotides at position 2 to 8 in a microRNA. So here we usually need a perfect base pairing to identify targets. And generally multiple sites are required for a strong repression. There's some kind of a cooperativity between different microRNAs, which we molecularly do not really well understand. One microRNA can control hundreds of mRNAs, and one mRNA can be a target of many microRNAs at the same time. So there is a lot of here combinatorial regulation. So these properties make actually microRNAs very suitable agents for networking and fine-tuning of gene expression because they can target several mRNAs in the same pathway, for instance, and usually their effects are small, 2 to 3-fold, but since the message can be controlled by many microRNAs, sometimes those effects are stronger. So they facilitate rapid switching to new developmental programs because these regulators acting as RNAs, they don't have to go to processing, transcription processing, translation. They can be made available faster than protein regulators. But there are also examples of microRNAs actually as work on or off switches of gene expression, and I will show you one example of this sort later. So this is microRNA field is a relatively young field. So first microRNA was discovered in '93, but then really the field kind of started in the year 2000, 2001, when a second microRNA, which turned out to be conserved between C. elegans and humans, was identified. This was LED7 microRNA. So there are many remaining challenges, and we actually have a lot of work still to do to identify mRNA targets and microRNA function in health and disease. Again, this prediction of targets because of this minimal complementarity to mRNA is not an easy task, and predictions can be have to be always supplemented by experimental evidence. So we also only start now to understand microRNA function at the organismal and tissue levels because most of the experiments there are done in tissue culture, but now people started to do knockouts and identify really true phenotypes of microRNA deletion. Well, I'm going to address these 2 points mainly, mechanism of inhibition of protein synthesis and regulation of microRNA biogenesis, microRNA repression itself, and mRNA turnover. So let me start with mechanism of inhibition of protein synthesis. We and others studied this since about 10 years ago, and we still don't really exactly understand how the microRNAs bring about the inhibition of protein synthesis. So certainly they inhibit translation and deadenylate mRNAs. But as to the translational inhibition, it is not really, most of the evidence indicates that microRNAs repress initiation at an early step, but there are also some data arguing that they could act as inhibitors of elongation. What is clear that most of the mRNAs which are subject to the repression, they are also partially strongly or less strongly deadenylated, and after deadenylation, messages are often degraded. So we have been working actually on the mechanism of translational inhibition over the years, and our data support inhibition of the translation step at the early initiation step, and for instance, we were observing polysomal shifts with both reporters and endogenous mRNAs, finding that repression is cup dependent, that translation from IRA's internal initiation site is not affected. Also, in the work done in partial collaboration with Nahum Sonnenberg's lab, we could show that in Krebs ascites extracts, the repression is cup dependent. But still, there are many issues which remain unresolved. Relative contribution of translational inhibition versus mRNA degradation. Why some mRNAs are not degraded at all, but others are very, very strongly affected. Stability of others is very strongly affected. Are the deadenylation and translational repression independent of each other, or deadenylation is a consequence of translational repression happening first? So I'll address this later. And also mechanism of the repression itself, how this initiation step is repressed. What factors are recruited by Argo and GW182 proteins, and how do they affect both processes? So I need to introduce 2 main players in a pathway. Those are proteins of Argonaute family. Argonaute proteins associate directly with microRNA, and there is some structural information available. The pockets which bind the 5' and 3' end of a microRNA have been structurally determined. There are 4 proteins in mammals, Argonautes 1 through 4, and these are multi-domain proteins with key domains being MEAT, PV, and PAS, which recognize different elements in a microRNA. And the second family of important proteins are GW182 proteins. GW stands for glycine tryptophan rich. There are 3 paralogs in mammals called TNRC6A, B, and C. And the function of algoprotein is actually to bring the GW protein to the complex. So and this interaction occurs between the mid-domain of Argonaute protein and GW repeats present at the N-terminus of this domain. So what happens is that there are layers of interactions. So microRNA actually brings algoprotein to the message. Algoprotein brings GW182, and then the question remains, so what is the next step in the inhibition? How translation is repressed, how the deadenylation is brought about. And I just want to show you a summary of the research done by us and also 2 other groups published recently. We could show that GW182 proteins through these regions recruit a large complex known as a CCR4-NOT complex, which actually is involved in both translational repression and mRNA deadenylation followed by degradation. So what is this complex? This is a highly conserved complex, conserved between yeast and humans, consisting of about 10 subunits. It contains 2 mRNA deadenylases, CAF1 and CCR4. It also has a role in transcription. One of the subunits, node 4, is a ubiquitin ligase, and clearly this complex has cytoplasmic and nuclear function. It is a very, very important complex with having major effects on gene expression, certainly very, very much understudied complex. But now, because of the connection with microRNA repression, people started to study it very intensively. So how is this complex recruited by GW182? I want to spend a few minutes on that because this is kind of interesting story. So as I told you, by deletion analysis of both Drosophila and human GW182 proteins, we originally identified 2 regions which are involved in the repression of protein synthesis by this protein. The N-terminal part called NED and C-terminal part CED. And by further mutational analysis, we could identify subregions in these 2 N-terminal and C-terminal regions which are key for the repression. And here there's some paradoxes, because this domain, RRM, RNA recognition element, this is the most conserved part of the protein in terms of the sequence, but this is absolutely unessential. PAM2 plays a supplementary role. What is important are the sequences in between called M2 and C-term, and on the N-terminal part, it's another region, and what the stars here indicate, here and here, are repeats of tryptophans followed or preceded by glycine, serine, or threonine. And these 3 regions are key for repression and CCR4-NOT binding. Those regions are disordered, so sequence prediction and also some NMR studies we did in collaboration find that there's no any structure in these regions. So they are both disordered, and these elements, tryptophan rich elements, they are redundant. So for instance, we can duplicate M2 sequence or C-term sequence and have the effect of the whole region. So as I told you, PAM2 has some stimulatory role. So I want to show you one example of the experiment how we prove that tryptophans dispersed through these non-structured regions are contributing to the repression. So when we take a wild-type sequence and tether it to mRNA, we see about 10-fold inhibition of protein synthesis. So from this level to that level. If we mutate single tryptophans, any of those, we don't see any effect. But when we start to introduce increasing amount of mutations of tryptophans, this plus this plus this plus this. So with 2 we see some effect, 3 more effect, and if we mutate 7 or 8, we completely eliminate the repression. And we also eliminate interaction with the CCR4-NOT complex. So those are just 2 examples of 2 subunits of the complex, CAF1 and C-NOT1. When we have wild-type sequence, these 2 proteins are pulled down. When we take away tryptophans, the interaction is gone. And the same applies to the NED domain. When we take the one which I was showing on a previous slide coming from the N-terminus, we find the same effect. Tryptophans are needed for both repression and CCR4-NOT binding. And what is most important, that these motifs also function in the context of full-length GW182 proteins and during genuine microRNA mediated repression, because many experiments which I was showing so far, they involve tethering of fragments or mutants of the protein to the mRNA, but by doing different genetic and biochemical tricks, we can knock down endogenous proteins and introduce mutants to the cell and show that activity depends of the reintroduced proteins depends on the tryptophan motifs. Of course, we were very much worried by these experiments that when we mutate 7 or 8 tryptophans, even in the non-structured regions, you can mess up this protein fragment quite a lot. So we were interested in doing kind of a gain of function experiment to show that those tryptophans are really not working by disturbing the secondary structure or folding of the protein, but that they genuinely play a role in the repression. So what we did, we decided to test whether any disordered protein from yeast, this was an example, will gain repressive potential upon insertion of tryptophan residues next to glycine, serine, or threonine. So here's a fragment of the yeast protein, SIC1, which is non-structured, so the structural studies were performed on this protein. And what we did here, we just inserted, shown in red, a couple of tryptophans into the sequence. Not really inserted, we introduced mutations creating GW or TW repeats in a protein. And you really gain the repressive potential. So when we insert 4, we see partial repression. When we insert 7, we have more repression. And what is even more important, when we insert those tryptophans, this protein, the control protein, doesn't pull down CAF1 and C-NOT1, does not interact with the complex, but the protein which have this tryptophan is inserted starts to pull down the protein, the CCR4-NOT complex. So this was a very important experiment for us. We actually repeated this with another independent yeast fragment which has no non-structured fragment and got the same result. So this tryptophan role is not really due to the disruption of the structure of the GW182 fragment. So the recruitment of the complex containing deadenylase subunits, of course, explain very nicely why the microRNAs and GW182s would induce deadenylation and mRNA decay. But then because those are catalytically active subunits, so this would explain why the protein is deadenylated. But does this complex also introduce translational repression of mRNA? And to study this, we turn out to the messages which do not contain poly A. So they contain either histone stem loop at the 3' end or the ribozyme, which cuts the message here. And these messages then can be studied using the tethering system, which I was referring to. So we can tether the proteins which are expressed as fusion with a lambda N peptide, which tethers the protein to the hairpins which are inserted to the 3' UTR. So we can tether AGO or GW182 or CNOT subunits or mutants thereof and check the effect on activity. And what we found is that tethering of the GW182 CED, this repressive fragment, but not of its mutant containing mutations in tryptophan residues, inhibits translation of poly A minus mRNA without introducing any degradation. Even more, we could demonstrate that when we tether the components of the CCR4-NOT complex, CAF1 or CNOT1 in S2 cells, we also inhibit activity of poly A minus mRNAs, either with a histone stem loop or of the ribozyme. And this repression is not associated with the degradation of mRNA, as shown here by the northern. So what we could conclude from that also these proteins induce genuine translational repression of mRNAs. Those are not the only experiments which support this conclusion. I have no time to show others. We also carried out some kind of epistatic analysis to show that actually GW182 protein functions upstream of CCR4-NOT. So we could show that repression by GW182 fragments depends on CCR4-NOT complex, but repression by CCR4-NOT complex components, like here, is GW182 independent. So we can knock down GW182 in the cells, and this repression will still be taking place. So this clearly puts CCR4-NOT complex downstream of GW182. GW182 attracted by AGO, then in turn attracts CCR4-NOT complex, which functions as a repressor. So just to summarize this part, the WGST motifs, we refer to them as W-motifs, tryptophan motifs of these proteins, represent key elements in introducing microRNA-mediated repression. And there are certainly two classes of these motifs in these proteins. One class, I was showing these elements, GW elements, at the very N-terminus of the proteins. They attract Argonaute proteins. They interact with Argonaute proteins. But then there is another class which interacts with CCR4-NOT subunits. We don't know how those motifs really work in CCR4-NOT recruitment. How is this additive effect explained? The most likely possibility is that there is a series of weak interaction between the tryptophans and maybe hydrophobic motifs in CCR4-NOT subunits. We know that this is CNOT1 subunit now, which interacts directly with the protein. It is probably unlikely that these motifs recruit several complexes, because those are quite big complexes, so it would be really difficult to accommodate them on such short piece of the protein. So there's some kind of analogy here maybe with nucleoporin importin interactions. You know that nucleoporins importin interactions are based on multiple phenylalanine glycine repeats, which work in an identical additive fashion as GW182 CCR4 interaction. We know that the protein which acts as a direct contact of GW182 is NOT1, It's the largest, about 2,500 amino acids long subunit scaffold of the CCR4-NOT complex, but how the complex actually represses translation, we don't know. So before I move to the final conclusion of this part, I just wanted to mention another another important question. What is regulating relative contribution of translational inhibition versus mRNA degradation? Are the deadenylation and translational repression independent of each other, or deadenylation is a consequence of translational inhibition? And there were already some indications in the literature. This is actually an experiment done in Nahum Sonnenberg's lab with some collaboration of ours, which demonstrated that in vitro, translational repression actually comes first, before deadenylation. So you see that maximal repression is already achieved after 1 hour, and if one looks at deadenylation, deadenylation is induced later. We then decided to look at this in human cell lines, in HeLa cells, by constructing inducible reporters and putting them into a specific locus in a HeLa genome and under control of inducible TET repressor. So we introduce into the same locus either control reporter, Firefly, luciferase, or Ranila luciferase reporter, which have a 3' UTR of the established targets of microRNAs, or mutated version of it. And we look at different targets containing either multiple sites or single microRNA site. And control Firefly receptors, they are expressed in the same way, independent if Ranila luciferase contain a mutant or wild-type 3' UTR, but when we have a wild-type 3' UTR, this UTR is strongly repressive. So here we compare a mutant, Ranila, which is not repressed by microRNAs, with a wild-type, which is repressed. We did many controls to show that this is due to genuine microRNA-mediated repression. And then we look at the kinetics after induction of expression of this gene by addition of tetracycline. So we see that already at earliest time points, we decrease protein expression, Ranila activity, but mRNA levels are decreased with a considerable delay. And this is seen with all 3 reporters which we were testing. And then we did a pulse chase experiment where we actually induce expression by addition of tetracycline, and then after 1 hour we add actinomycin D or alpha-amanitin to block POL2 transcription and look at mRNA levels, poly A status, and protein levels. So again, we see that at earliest time already, we see protein synthesis repressed without any effect on mRNA level or poly A length, and only at later time points we see some effect on poly A length, this is this, and mRNA level, and eventually after longer time the repression can be explained by mRNA degradation in this case. So we can interpret these results as indicating that actually translational repression comes first. And after with some delay, 20 to 40 minutes, the deadenylation is taking place, and this is followed by mRNA decay. And similar results were recently, this is in the last issue of Science, published for zebrafish by Antonio Giraldez's lab and flies by Rachel Green's lab. And altogether, they indicate that translational repression is probably the dominant factor in microRNA function at early time points. This is, in a way, an important point because there were several papers, some of them coming from David Bartel's lab at MIT, arguing that actually degradation is explain everything, and the kind of default function of microRNAs is mRNA deadenylation and degradation. And this is actually hard to understand that this would be the case because there are many examples in the literature coming from not genomic studies where one is looking globally at the whole genome, but in individual mRNAs when one is seeing mRNAs which are repressed by microRNAs at a translational level without considerable degradation of mRNA. And here it's probably important to mention now that perhaps the fact that the same complex, CCR4-NOT, is responsible for deadenylation of mRNA and translational repression makes one to speculate that depending on a cell type or microRNA or a message, either activity of this complex to deadenylate or repress translation will be a dominating one, and one could again go back and forth between these two activities of the same complex. So this is just what I've been discussing. Another point which I want to make is that these layers of interactions above, the microRNA attracting AGO attracting GW182, GW182 attracting CCR4-NOT, and who knows how many factors are working in between, creates a lot of possibilities to regulate whole process. And if one thinks about microRNA regulation as a pathway affecting more than half of the genes in a cell, this is probably not unexpected that this complexity subject to post-translational modification and proteolysis is there actually to allow kind of graded stages of inhibition. So I would like now to move to the second part, to regulation of microRNA-mediated repression and metabolism. So I only very briefly want to mention the work which we did on the regulation of microRNA repression per se. There are dozens of regulatory proteins now identified which modulate the repression by microRNAs. Factors which associate with AGO GW182 proteins bind next to microRNAs at mRNA. There's also emerging role of post-translational modifications. So we still don't understand really biology, but all these proteins are post-translationally modified, ubiquitinated, hydroxylated, or phosphorylated, and probably this plays an important role in the regulation. And finally, the reversibility of microRNA repression. At one point, a couple of years ago, when we started to do these experiments, it was only known that microRNAs bind to mRNA and repress mRNA as a kind of final the decision. And especially when microRNA was degraded, that was, things were over. So we reasoned that the process would be actually much more important, much more dynamic, when under some conditions, microRNA repression could be reversed, and you could go back and forth. And indeed, we found that in hepatocytes, the microRNA 122, which is a very abundant liver-specific microRNA, it represses mRNA encoding cationic amino acid transporter by binding to 3' UTR, and the repressed mRNA is targeted to P-bodies for storage without any degradation, endogenous mRNA. And then we noticed that upon stress, this can be ER stress, amino acid starvation, or oxidative stress, HUR, an RNA binding protein, is getting out of the nucleus, binds to the 3' UTR, and it causes relief of the repression. So mRNA is departing from the P-bodies and recruited to polysomes for translation. And just want to show one example of the data. So here in a cells grown in the presence of amino acids, the repressed mRNA is present in P-bodies. When we starve cells for amino acids, the P-bodies are still there, but mRNA is gone, and it is associated with polysomes. So this process is HUR dependent, so when we knock down HUR, this doesn't happen. And treatment with anti-miR122, antisense oligo, actually is mimicking this phenotype. So this is indicative of really microRNA repression being relieved. And HUR, to introduce you briefly to this protein, you are probably familiar with it because Miriam Gorosbe, who is at NIH but not at this campus, is studying this protein very intensively, is an RNA binding protein which binds to ARE elements, AU-rich elements in 3' UTR, and usually stabilizes mRNAs. It's mainly localized in the nucleus, but shuttles between the nucleus and the cytoplasm and translocate from the nucleus to cytoplasm in response to stress. And this protein is upregulated and accumulates in the cytoplasm in many cancers. So we could actually demonstrate that we can mimic this protein, the activity of this protein in the absence of stress. So we can uncouple this effect from stress because originally it was very hard to predict what the stress conditions, what kind of changes they would be inducing in a cell. So by using HUR mutants in cancer cells with cytoplasmic HUR, we could show that cytoplasmic location of HUR is actually sufficient for the effect. And more recently, we were interested in the mechanism, how this protein actually is mediating this effect. And we could show that actually this protein, when binding to 3' UTR, displaces the microRNP from mRNA, and it does it because it oligomerizes along the message. And how exactly does it physically displace microRNA? This we don't know. I have no time to show this data. They were recently published by Pradipta Kundu, a student in a lab. So I want to finish this part with just telling you that this interplay between microRNAs and RNA binding proteins, binding to different regions in a 3' UTR, is a very common phenomenon. And there are dozens of examples of this sort described. Ours was the first, and this can work in different directions. RNA binding proteins can have a positive effect on repression and negative, and vice versa. MicroRNPs can have a positive or negative effect on RNA binding proteins, and this can be due to different mechanisms. Very often, microRNAs and RNA binding proteins just compete for the same sequence to bind to. So since 3' UTRs, again, are very often very long, and there are hundreds of RNA binding proteins and hundreds of microRNAs, you can imagine how common this type of co-regulation will be. I want to then move to the regulation of biogenesis, just by telling you that there are many factors which modulate biogenesis of microRNAs at the step of Drosophila processing or dicer processing. And these proteins include proteins like P53, SMAT and HNRP proteins, KSRP, P68 helicases, Lin20, 28, and oncogene. So there are many, many examples of these proteins binding to the usually hairpins of microRNA precursors, either in the nucleus, and then they affect Drosophila processing in a positive or negative way, or in a cytoplasm where they affect in a positive or negative way processing by dicer. This is taken from an article published some time ago in collaboration with Roberto Gerzi and Michael Rosenfeld. I want to now just move to the final piece of experiments I want to discuss, the regulation of microRNA decay. MicroRNAs are generally believed to be very stable molecules, very long-lived, with their half-lives extending into days even. There are examples of microRNAs in heart or liver which do not change their levels over days. So we actually started to investigate this problem in retina. We were interested, I have a very good colleague, a specialist and retina physiologist in the institute, Boton Troska, and we decided at one point to combine forces and look at microRNAs which respond to light or dark adaptation. So we did a set of experiments where we're keeping mice in light, or moving them after 3 hours in light into dark, and collecting samples of mice kept in light or in dark, and doing deep sequencing, RNase protection, real-time PCR, microRNA arrays. And we came up with a couple of microRNAs which actually decreased during this 3 hours in dark about 2.5 fold, which was something unheard of, that microRNA would be going down so rapidly at that time. So this was done a couple of years ago and published recently, but now there are more examples of microRNAs actually turning over faster. So this is just RNase protection assay showing the same. So we then did a kinetics collecting retinas at half-an-hour intervals, and we found that actually this decrease is never more than about twofold, and it's relatively rapid with half-life of about 45 minutes. And by putting the animals back to light, there was a very rapid increase in microRNA level. So by doing a lot of biochemistry and inhibitor studies, we could show that in dark, microRNAs generally decay very rapidly, and transcription of these microRNAs which are regulated is low. Now you move animals to light, transcription is up, and this compensates for the decay. But we were surprised to find out that even microRNA level of each is not regulated, like LET7, is also very unstable. It has the same levels in dark and light, but this is due to high transcription and rapid decay, which compensate for each other. So this is just an example of the experiment where we inject alpha-amanitin to one eye, using second eye as a control, and we show that LET7 microRNA, the one which was not changing at level, is going down when you inject alpha-amanitin, and decays very, very fast. So this, and we found similar results for several other microRNAs which do not go up and down in dark, but they decay upon injection of alpha-amanitin. So what happens in the retina is that microRNAs turn over by default very fast, and only when you need to increase microRNA level in light, you increase transcription. When you put the light off, transcription is down, and then of course the level of this microRNA goes down. And we actually have some biological understanding what is going on. One of the targets of this cluster, which changes the level in dark it goes down, controls expression of a glutamate transporter. And in dark, there's a continued release of glutamate into the first visual synapse. And you need to update this glutamate very fast, because otherwise too high levels would be toxic, and you induce in dark expression of this particular glutamate transporter. Now the question is, so this is in neurons. We then tested also primary retinal glial cells, Müller cells from retina. There's no turnover. Retinal pigmented epithelium, no turnover. ES cells, NIH3 tissues, no turnover within 10 hours or so. So the question, and this is my last question I want to briefly discuss, do microRNAs turn over also rapidly in non-retinal neurons? Yes, they do. And we looked at hippocampal slice cultures, organotypic cultures, hippocampal and cortical neurons, and also neurons derived from ES cells, and looked at different microRNAs, neuron-specific, glia-specific, and others. And you can see that when we look at organotypic hippocampal slices, the neuronal microRNAs go down when we block transcription with alpha-amanitin. When we look at hippocampal structures or neurons or cortical neurons, we see the same. Again, they go down about twofold, but only twofold. What was the most interesting, perhaps, that this turnover is entirely dependent on neuronal activity. When we block action potentials with tetrodotoxin. We block this turnover, and we did controls where we were looking at the levels of mRNAs or primary transcripts of microRNAs. Those are not affected by this. So you need firing neurons and active neurons for turnover to take place. And we extended this to also ES cell-derived neurons and hippocampal neurons, looking at the effect of glutamate or blocking glutamate receptors. When we add additional glutamate to neuronal culture, we actually accelerate turnover from this rate to this rate. And the same is true for hippocampal neurons. When we block kinase or AMPA receptors with different chemical compounds, we block turnover. We also have one microRNA which behaves in a completely opposite way. Its turnover depends on blocking activity, and this microRNA actually has opposite function to other microRNAs in synaptogenesis and dendritic spline development. So here is just my last discussion slide. Why microRNAs would turn over so fast in neurons, and why would it depend on neuronal activity? This decrease in about 50% level makes us to think, and we have some preliminary data supporting this, that only one pool of microRNAs is turning over. And perhaps the one which is present at synapses in dendrites is actually turning over fast. There is a recent paper in Nature Neuroscience indicating that BDNF actually is having an effect at the synapse on a microRNA turnover. So possibly it is well known, well established that translation is regulated at the synapse, and this activation of translation at the synapse is needed for connectivity for long-term memory formation. So perhaps the stimulation and activity increase of translation at the synapse is associated with the removal of microRNA, which is active as an inhibitor of translation at the synapse, and there's a lot of support for this idea already available. So an alternative possibility would be that you need the stimulation of neurons actually eventually is activating also transcription in the nucleus. And perhaps you need to turn over microRNAs in a cell to make new microRNAs available for loading into newly transcribed messages, which then travel from the nucleus to the synapse. So this is all kind of hand-waving stimulations. We have no idea, but we are actively working on this. We don't know what is the mechanism of turnover. We were looking at the levels of components of microRNA machinery like dicer protein or agoproteins. Those do not change. There's probably some kind of induction of nucleases which are degrading the mRNA. I think I only want to now acknowledge my collaborators. So those shown in red here are contributed in a different way to this part. I only want to list in particular Jacek Kroll, who did retinal work with Boton Trowska and his group, Marina Czekulayeva and Hans-Rudi Matis, who did GW182 work, and Pradipta Kundu and Julien Betounet, who did HUR work and these kinetic experiments, respectively. We collaborate with many people, particularly with Nahum Sonnenberg, Gerhard Schratt, and also other people. And I don't know whether I really mentioned this, but the way that we are not the only ones working in this field. This is a very competitive field, and the many studies done by Nahum Sonnenberg's lab and Isao Rauda actually were often coming to similar conclusions, which is actually very good because there's a lot of conflict in microRNA studies. You know, one group cannot confirm the results of other groups. So the fact that 3 groups identified CCR4-NOT complex as a downstream component is something unheard of in microRNA research, what makes us actually very happy about it. Thank you very much.
So I think we have a few minutes for questions. Please, if you would like to ask a question, go to the microphone.
So, very nice talk. You showed you provided evidence that it's translational repression followed by mRNA degradation, and I believe the mRNAs that you are looking at and the microRNAs were basically targeting the 3' UTR. But it's also known that some microRNAs can inhibit gene expression by exclusively binding to the coding region. So do you think that a similar mechanism would be applicable to those targets that are regulated by binding of microRNAs to the coding region?
We did not address this ourselves, but Mihaela Zavolan, who is a bioinformatics professor, she analyzed genomic and CLEAP data of others, and she concluded that actually when sites are located in the coding regions, there is less effect on degradation but more effect on translation proportionally. Although generally the sites in the coding regions have less effect, cumulative effect than sites in the 3' ETR regions. So there is a difference there actually.
It was a great talk, Vitek. I had two questions about the first part. So when you're using the mRNAs that lack the poly A tail and you still see the translational repression, are there any clues about which subunits of the CCR-NOT complex, say in knockdown experiments, are required for that repression, besides not one, which you would be?
Yeah. So not one, of course, is essential because it's essential for the assembly of the complex. So the suspect actually is CUF1. There are some data from Marvin Wickens' lab who was tethering CUF1 in xenopus oocytes, and he finds that if CUF1 is tethered to even poly A minus mRNA in injected oocytes, it inhibits translation in a CUP-dependent way. There is a lot of kind of circumstantial evidence that CUF1 might act as an inhibitor of translation or translation initiation in yeast also, coming from different groups, which makes us thinking about CUF1 as a possible factor mediating this. Although if one looks at CUF1, at least in mammals, they have practically nothing apart from catalytic domain of deadenylating enzymes. So we are looking into that. We don't know. We don't know.
Okay. The other quick thing I was wondering about is the GW-repeats, they contain serines and threonines. So it sort of sounds like the POL2 CTD. Is there any evidence for phosphorylation of those serines and threonines that might affect the interaction?
So we did not look ourselves, but in a mouse proteome databases, there are some phosphorylation sites identified in GW182 proteins. None of them falls into the serines or threonines which are next to tryptophans, which we are looking at. This was, of course, a very attractive idea because that would perhaps modify accessibility of the repeat to CNOT1, but we have no evidence of that.
I have a question regarding turnover of microRNAs. So in plants, there's been a couple of recent studies that there is a machinery which deadenylates microRNAs, and that is important or linked to their turnover. I was just curious if something similar happens in mammalian cells.
Yes. So there is a link. So there are some studies coming from Phil Zamore's lab indicating that when microRNA is base paired to the target with high complementarity, so resembles siRNA rather than microRNA, it is uridylated to the 3' end and then degraded. So uridylation might be an intermediate step in degradation of microRNA by a degradation machinery. We don't know how it is in neurons. We were actually doing deep sequencing on microRNAs, under conditions where you have turnover, you don't have turnover, but we didn't get a clear answer from that.
You know what's the nuclease that degrades them? Is it exosome or some other nuclease that degrades those RNAs?
Okay, so far there are two classes of enzymes implicated in the degradation. In plants, those are 3' 5' exonucleases, called CED1, CED2, CED3. I think you have to knock them down, three of them, to see the phenotype. In mammals, no, there's no work in mammals, but there is a work done in C. elegans, which implicates actually XRN1 and XRN2 in a 5' to 3' direction degradation. So they're completely opposite enzymes with opposite activity implicated. We don't know what it is actually in mammals.
Thank you.
So there will be a reception right next in the library. Everybody is welcome, and thank you very much.
Oh.